Good afternoon. This is Miss Dagenford, and I wanted to give you a sense of where we're going to be going next with our DNA. We in class the other day, we were able to go over Fred Griffith and Hershey and Chase and Avery McLeod. So we've gone over some of the experiments, and one thing that we've gotten to is the fact that they know that DNA was the transforming principle, and now we know that DNA is the genetic material. But then the race was on to figure out, what is DNA like? What is DNA the shape? How does it work? Well, part of what you need to understand is the two contributing scientists. And there were more than that, but these are some of the big contributing scientists. So, one of the objectives you should be able to do now after this video, you should be able to describe DNA experiments and their significance. And we're also going to be looking at DNA structure. So, one of the first scientists, and you saw a little bit of the video with her, is Rosalind Franklin. And Rosalind Franklin was really big into X-ray crystallography. And X-ray crystallography is, just as the name implies, you're able to get a molecule to the point where you can bombard it with X-rays, and you get a scatter pattern. And as the X-rays are deflected, it'll expose a film, and that will allow researchers to know what the molecule is shaped like. And she did this with DNA, and she came up with a very famous photo 51. And photo 51, what was a big hallmark realization is this helical shape, a spiral. And this was very, very helpful in retrospect. Everybody kind of realized this was the picture that helped them discover that it was a double helix. That's what really helped. So it was a repetitive structure, as a helical structure. There were a lot of competing theories. Was it a triple helix? Was it a double helix? But we know now, and they figured out it was a double helix. Then the question was, well, how are the bases arranged? Are they in the same? Like, is each helix the same? So is one side of the strand the exact same as the other side of the strand? Well, part of the solution came from a scientist whose name was Erwin Chargaff, and his big insight was that there always was the same percentage of adenine and thymine. Now, before you hit your questions and think, well, what in the world is she talking about? Adenine or thymine are two of four crucial chemicals in DNA. And they realized that every time they saw an adenine, there was a thymine. And then it was the same thing with guanine and cytosine. Every time they saw a guanine, they saw cytosine. So just to give you some examples, and this can be different between species. So each organism had a different combination. Now, these numbers are not accurate. Don't go looking it up. It's probably not the right number. But for instance, you could have a dog where if we had 20% adenine, we would see 20% thymine. Now, it wasn't a perfect. Oftentimes they might have like 19% or 19.8%, but it was that idea. And then if you had 30% guanine, you had 30% cytosine. And then if we went to a chicken, you might see 23% and 23%, or 27 and 27. And of course, they have to add up to 100. So, same thing with the banana. So, let me just give you one to play around with, and then I'll ask you a question. What if we took a sparrow, funny little bird, and let's say the sparrow had 21%? Well, how many T's would we have? And then also think about how many G's and C's would we have. It's just a simple math question. Take a look at the other ones and see what you can do. Now, Watson and Crick were the two scientists that took all of the information and they were able to put it all together and had the insight to kind of come up with that model that you see in this picture. Very famous picture of them looking at this model that they've made. And they created what is known as that double helix model. They were able to figure out all of the pieces of the puzzle and put it together. So what do we mean by the structure of DNA? Well, let's take a look at the picture in the next. Now, what we want to start with is the DNA structure. Well, first of all, let's lay down some objectives. by the end of this video, you should be able to describe that organizational structure. How is DNA organized? And then to identify those base pairs, and then to explain DNA replication. That's our goal. So let's go ahead and tackle the base pairing rules. You're going to see that DNA, every time you see an adenine on one side of the DNA, you're going to see a thymine on the other. They pair together. If that's not immediately clear right now, it will be. You'll see pictures and you'll do the aha moment. Same thing with cytosine and guanine. Every time you see cytosine on one side, you will see a guanine on the other side. Vice versa. If there's a guanine on one side, you'll see a cytosine on the other. And they do this because of the chemical shape that they have. You'll see pictures in a second. And what holds them together are hydrogen bonds. And just a quick review of hydrogen bonds. Hydrogen bonds exist between an atom that has a partial negative charge and then another atom that has a partial positive on another molecule. So just as a quick reminder, one water molecule, that's our partial negative, that's our partial positive, and the red lines are our hydrogen bonds. And if you recall, they're designated by our little dotted lines. So what does DNA look like? Well, let's take a look at these three models. Over on the left is the picture that if you looked at Watson and Crick's original paper, this is what they kind of drew up. And it's a nice standard model. We still use it. We have 10 bases. You can count it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 bases per turn, full turn, and then you see the two strands spiral around each other. So that's kind of spatially what it looks like. And then in the middle, you see the arrangement. So this is what I mean by how it's organized. So let's take a look at the backbone. So we're going to see one backbone over here, and then one backbone over on the left here. What I mean backbone, like the sides of the ladder, like what creates the outside edges. So we have a phosphate. This is a phosphate. And then a sugar. And the sugar in this case is deoxyribose. And of course, hit pause if you ever need to just stop and make sure you write down and label. And notice it just alternates that way. Phosphate, and then deoxyribose. Phosphate, deoxyribose. Phosphate, deoxyribose. And then connected to the deoxyribose would be one of 4 different bases, nucleotide base, nitrogenous bases, pardon me. And so we have thymine, guanine, cytosine, adenine. And notice where there's a thymine on one side and adenine's on the other. Guanine, cytosine, cytosine, guanine, adenine, thymine. Now, one thing you might notice if you're very observant, one strand, the little oxygen here is going this way. The other side, the right side, is going where the oxygen is pointed down. We're not going to worry about that too much. This is still an introductory class. So let's do a quick practice. If we had our phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, PD. And so we could make the other one, and if I wanted to be really accurate, I could put them upside down, but that might confuse the issue. So phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, PD, PD. My strand's going in there a little bit. And attached to every deoxyribose would be a base. So let's just make it up. A, T, G, G, A. Now, they can repeat each other, right? You can have A. What would the other side be? Just pause it for a quick second and think, what would the other strand of DNA be like? And hopefully you said T, A, C, C, T. And that would be our base pairing rules. That would be a nice example of that. Now, between these, we'd have our hydrogen bonds. Between A and T, we see 2 hydrogen bonds. Between G and C, we have 3. And you can see in this picture, they use dotted lines. The central picture, they represent dotted lines. Mine, I can't do dotted. I'm putting a dash there. We'll talk about why that might have an effect on DNA in just a bit. Now, so just to look at these a little bit closer, the two bases, adenine and guanine, notice they have two rings. They're called purines. Now, I did this back in college days. I came up with a stupid little name to help you remember. Purines, pure, and then AG. Purag. That helps me just to remember, okay, those are the two bigger ones, adenine and guanine. And then pyrimidines are going to be thymine, cytosine, and uracil. We'll get to that when we get to RNA. So purines are going to be the two double ones, and then pyrimidines are the single ring. And so in that regard, you know that A and T, that's a double and a single. G and C, double, single. And DNA, we knew that DNA had a uniform width. That was also another reason we understood how that worked. So here's just another picture. There's our 2 hydrogen bonds and 3 hydrogen bonds. Now this picture I like because it shows you those hydrogen bonds. Here's our partial positive. Here's our partial negative. And then just another picture to kind of represent it. You can see here, I'm going to pause in just a second. You're going to be asked a question. Here's one that's double. Here's one that's single. And then just this last picture, I'm going to pause here. I'm going to zoom out just for a second. You can see all of these different, here's our, the P and the D that I was talking about earlier. Phosphate sugar, phosphate sugar, phosphate sugar, and the sugar is deoxyribose. And then here's our sugar, phosphate, sugar, phosphate, sugar, phosphate. Here's our nitrogenous base, and then the little dotted lines. So notice I have over on the side, here are some words that you can match up with, and that'll help us to end the video. When we come back, we want to talk about DNA replication and move on from there. I hope you had a great Thanksgiving holiday, and I shall see you when you get back.